your parents, you can see the features that you share with them, such as the shape of the eye, the presence of your dimples, or even the hands that you use to write. The sharing of features can be explained by heredity, where traits are passed from parents to offspring. Yet, when you look at your brothers and sisters, each one of you can be considered unique on the combination of traits each possesses. That is variation, which demonstrates differences among individuals. On this video lesson, we will explain inheritance based on Mendelian laws, and we will solve monohybrid and dihybrid crosses given the phenotypes and genotypes of the parents.
Let us have a review of what we have learned last time. True or false? During interphase, the cell grows, replicates its DNA, and prepares for mitosis. If your answer is true, then you are correct. Next, mitosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. If your answer is false, then you are correct. Next, we have synapsis takes place during prophase 1 of meiosis. The answer is true. Next, we have homologous chromosomes are 2 pieces of DNA within a diploid organism which carry the same genes, one from each parental source. The answer is true. And gap2 phase is the phase of the cell cycle when DNA packaged into chromosomes is replicated. If your answer is false, then you are correct. Why the statement is false? Because during synthesis phase, DNA is replicated. On this video lesson, we will predict the phenotypic expressions of traits following simple patterns of inheritance. So this is the first part of the video lesson about the Mendelian patterns of inheritance. Make sure that you have watched the video lesson about hereditary characters. The link is flashed on the screen. So take note of these words that we will encounter as we go along discussing this topic. So we have genetics, allele, gene or genes, phenotype, genotype, homozygous, heterozygous, dominant, recessive. We will also include the words monohybrid cross and dihybrid cross. Genetics is the branch of biology that deals with the study of heredity and variation. Heredity refers to the transmission of traits from one generation to the next, while when we say variation, it deals with the differences among individual organisms. It explains why closely related individuals resemble each other. It also describes the processes involved in the development of new characters that explains why members of the same species show variations in the characters they express. Modern genetics began with the work of Augustinian friar Gregor Johann Mendel. Gregor Johann Mendel, which is an Augustinian monk or friar and botanist, is considered as the father of classical genetics. He was born in Heisendorf, Moravia in Austria, which is now part of the Czech Republic. In 1844, he joined the Augustinian monastery of St. Thomas at Prun, Austria, now Brno, the Czech Republic, at the age of 22. Like many of us, he also wondered how traits are transmitted from parents to offspring. He spent 10 years of his life experimenting on garden peas and another 2 years analyzing his result. Mendel experiments include studying the traits in peas. Mendel studied the inheritance in peas or Pisum sativum. He chose peas because they had been used for similar studies and are easy to grow and can be sown each year. Pea flowers contain both male and female parts, called the stamen and stigma, and usually self-pollinate. Self-pollination happens before the flowers open, so progeny or the offspring are produced from a single plant. Another reason why Mendel selected garden peas for his experiments is because the plant has several characters that exhibit two contrasting expressions or traits. Another reason for Mendel's choice is that garden pea plants reproduce at a fast rate. Thus, a number of generations can be produced within a short period of time. They are also hardy plants. They do not need much caring and cultivating. Moreover, garden pea flowers are structurally adapted for self-pollination, so they have boat-shaped keel petals that enclose the stamens and pistils. Peas can also be cross-pollinated by hand, simply by opening the flower buds to remove their pollen-producing stamen and prevent self-pollination, and dusting pollen from one plant onto the stigma of another. So therefore, cross-pollination can be done by manually removing the stamens and introducing the pollen of flowers from other garden pea plants. Mendel's experimental design includes three important steps. Number 1, Mendel studied one character with two contrasting expressions at a time, say, seed color, yellow and green. He produced pure breeding yellow-seeded plants by allowing the plants to self-pollinate for several generations. This ensured that all the pea plants that he cultivated for the experiment produced only yellow seeds from generation to generation. He followed the same procedure to produce pure breeding green-seeded plants. So when we say self-pollination, it is the pollination of a flower by pollen from the same flower or from another flower on the same plant. So two types of self-pollination. We have type 1, wherein it occurs with the same flower. Pollen from the anther is transferred to the stigma of the same flower. For type 2, it occurs between two flowers of the same plant species. Pollen from the anther of one flower is transferred to the stigma of another flower in the same plant. Number 2. Mendel cross-pollinated the two pure breeding plants with contrasting expressions for the same character. How did he do this? He removed the stamens of the green-seeded plants and brushed their stigmas with pollen obtained from the yellow-seeded plants. He also removed the stamens of the yellow-seeded plants and brushed their stigmas with pollen obtained from the green-seeded plants. When we say cross-pollination, it is the transfer of pollen from the anther of one flower to the stigma of another flower on a different individual of the same species. Mendel followed the inheritance of 7 traits in pea plants, and each trait had 2 forms. He identified pure breeding pea plants that consistently showed one form of a trait after generations of self-pollination. So we have here for flower color, we have purple and white. For flower position, we have axial and terminal. Seed color, yellow and green. Seed shape, we have round and wrinkled. For pod shape, we had inflated and constricted. For pod color, we have green and yellow. For the stem length, we have tall and dwarf. So every time that he crossed these pure breeding plants, he came up with results such as when he crossed the pure breeding pea plant with purple color with a pure breeding plant with the white color, the offspring produces purple colors, and so on, just like what is flashed on the screen. For number 3, Mendel allowed the F1 plants, or the first filial generation, or now considered as the parental generation 2, to self-pollinate. He counted the number of offspring, which is F2, that express each of the two contrasting traits for all 7 characters studied. From the P2 process or parental generation 2 process, Mendel made 2 important observations. 1. When the F1 plants that express only the dominant trait were self-pollinated, some of their offspring, or F2, expressed the recessive trait. The ratio of the plants with dominant traits to the plants with recessive traits is almost 3:1. Dominant and recessive traits exist when a trait has 2 different forms at the gene level. When we say dominant trait, it is the trait that first appears or is visibly expressed in the organism. For recessive trait, it is a trait that is present at the gene level but is masked and does not show itself in the organism. From the observations, Mendel concluded that the F1 plants must have a pair of unit determiners, one for the dominant trait and another for the recessive trait. For example, the F1 plants with yellow seed color have 2 unit determiners, one for yellow and another for green seed color. While the determiner for green seed color is present in the F1 plant, the trait was masked or dominated by then for yellow seed color. In 1903, American scientist Walter Sutton coined the term genes to refer to these unit determiners of heredity. A gene is the basic physical and functional unit of heredity. Genes are made up of DNA. The Mendelian principles of inheritance. To make it easier for Mendel to analyze the results of his experiments, Mendel decided to use letter names for the genes that control the hereditary character. Since each hereditary character has 2 contrasting expressions, he decided to use capital letter and a small letter to distinguish between the 2. For seed color, we will use the following: capital letter Y for yellow, small letter y for green. When writing a genotype, the dominant allele is usually represented by a capital letter, while the recessive allele has a lowercase letter. Capital letter Y and small letter y, or Y and Y, are called alleles, or different forms of the gene for seed color. An allele is one of 2 or more versions of a gene. Recall, though, that Mendel concluded that these genes appear in pairs. Hence, it will be more appropriate to represent the 2 contrasting expressions in each plant as follows. For yellow-seeded plant, we will use 2 capital letter Y. For green-seeded plant, we will use 2 small letter y. Yellow and green are physical expressions of the character for seed color and are called phenotypes. Capital letter Y and small letter y and y are genotypes which describe the genes of the individual. Phenotype is the individual's observable traits such as height, eye color, and blood type, while genotype is the organism's complete set of genetic material, or the genetic makeup of an organism. Gene pairs are described as homozygous if they are made up of identical genes. Pure breeding plants have homozygous gene pairs for character or interest. Hence, Mendel's pure breeding yellow-seeded plants have genotype capital letters Y and Y, and the pure breeding green-seeded plants have genotypes small letters Y and Y. Gene pairs that are made up of dissimilar genes are described as heterozygous. Homozygous means having 2 identical alleles of a particular gene or genes. Let's say homozygous dominant or homozygous recessive. Heterozygous having 2 different alleles of a particular gene or genes, 1 dominant allele and 1 recessive allele. When the gene pair is heterozygous, which one of the 2 genes will become physically expressed? A according to the Mendel's law of dominance, when the gene pair of an individual is heterozygous, only one of the two genes is physically expressed. That which is expressed is dominant, while the other that is hidden or not expressed is recessive. Let us cross pure-breeding yellow-seeded plant and pure-breeding green-seeded plants. First parental generation is we have yellow-seeded, which is pure-breeding, and green-seeded. Our gametes are, we have an allele for yellow seed, another allele for yellow seed, an allele for green seed, and another allele for green seed. For our first filial generation, so we will use the FOIL method. So first we have the genotype with an allele for yellow and green, another genotype with an allele for yellow and green, and so on. So our genotypes are as follows, and our phenotype is all yellow-seeded pea plant. The F1 generation produced from the P1 cross has a genotype of capital letter Y and small letter y, and the phenotype yellow seed color, hence yellow is dominant. While the gene for green seed color is also present, the trait is recessive, that is not physically expressed. We call this as a monohybrid cross, which is a cross between a pea plant that is homozygous for yellow seeds and a pea plant that is homozygous for green seeds. Will produce only yellow heterozygous offspring. So the first type of cross that we have, so this one, we call it as FOIL method, while this one is called as the Punnett square. The Punnett square is a square diagram that is used to predict the genotypes of a particular cross or breeding experiment. It is named after Reginald C. Punnett, who devised the approach in 1905. The heterozygous yellow parents with genotype capital Y and small y can produce two kinds of gametes, a yellow-seeded gamete or capital letter Y and a small letter y or green-seeded gamete. The cross between two parents with heterozygous yellow seeds produce a genotypic ratio of 1 homozygous dominant yellow, 2 heterozygous yellow, and 1 homozygous recessive, and a phenotypic ratio of 3 plants with yellow seed color and 1 plant with green seed color. Mendel then allowed the F1 generation to self-pollinate. From the result of the cross, where the F1 is now the second parental generation or P2 generation, Mendel formulated his second law, the Law of Segregation. The Law of Segregation states that a diploid organism passes a randomly selected allele for a trait to its offspring, such that the offspring receives 1 allele for each parent. This explains how genes, which control the expression of hereditary traits, are transmitted from parents to offsprings. So let us have another test cross for second parental generation. So we will use the heterozygous individuals or heterozygous yellow from the first filial generation. So the possible gametes are as follows. We have an allele for yellow and an allele for green, and another allele for yellow and another allele for green. So for our second filial generation, we have homozygous dominant, heterozygous, another heterozygous, and the homozygous recessive. For our genotypes, we have the following. So we have 1, 2, 3, and 4. For our phenotype, we have yellow, which is homozygous, heterozygous yellow, and green. Take note that the physical manifestation of traits of these 3 individuals are the same, which is yellow. However, their genotypes or their genes are not the same. One is homozygous and the remaining 2 are both heterozygous. For our genotypic ratio, we have 1:2:1. 1 homozygous yellow, 2 heterozygous yellow, and 1 green. For our phenotypic ratio is 3:1. Why 3:1? It is because these homozygous and heterozygous yellow are both yellow. Their physical manifestation of their trait is the same, and we have 1 green. The second parental generation, or P2 cross, self-pollinated in Mendel's experiment. Crossing two pea plants that are heterozygous for seed color will produce offspring in the ratio shown in the Punnett square. So we have 1/4 homozygous dominant, 2/4 heterozygous, and 1/4 homozygous recessive. Because the P1 and P2 crosses in this discussion consider the inheritance of only one character, seed color, they are called monohybrid cross. A monohybrid cross is defined as the cross happening in the F1 generation offspring of parents differing in one trait only. Let us have a sample problem. Widow's peak is dominant over straight hairline. Given that the mother has heterozygous gene pair for widow's peak and the father has straight hairline, what is the genotype of the mother? Number 2, what is the genotype of the father? Number 3, how many kinds of gametes can the mother produce? List them down. Number 4, how many kinds of gametes can the father produce? List them down. 5, perform a cross using Punnett square. Number 6, give the genotypic ratio of the cross. And number 7, give the phenotypic ratio of the cross. You may pause this video so that you can complete all the answers that is asked. Alright, let us compare our answers. For number 1, the genotype of the mother is 1 allele for widow's peak and 1 allele for straight hairline, or capital letter W and small letter w. Remember that widow's peak is represented by capital letter W since it is a dominant trait, while straight hairline is represented by small letter w since it is a recessive trait. The father's genotype is written as 2 small letter w since straight hairline is a recessive trait. How many kinds of gametes can the mother produce? The answer is 2. 1 allele for, or 1 gamete, or we call it a gene for widow's peak, and another allele for straight hairline. Whereas the father's gametes are, or is, the father's gamete is just 1, which is for the straight hairline. Next is perform a cross using the Punnett square and give the phenotypic and genotypic ratio of the cross. So let us watch this video.
Widow's peak is dominant over straight hairline. Given that the mother has heterozygous gene pair for widow's peak, the father has straight hairline. The genotype of the mother can be written as follows: 1 capital letter W and 1 small letter w, while the genotype of the father can be written as 2 small letter w. Why letter W for this trait? It is because widow's peak is considered as a dominant trait, and in preparing a key, we use the first letter of the dominant trait, so capital letter W to represent widow's peak since it is a dominant trait. And for the straight hairline, we will use small letter w since it is a recessive trait. The next thing that we have to prepare is the Punnett square. All you have to do is to draw a box and divide it into 3 columns and into 3 rows. Then the uppermost left side of the box, you have to divide it into 2 for the allele of the father and the alleles of the mother. For the mother, we have an allele for widow's peak and an allele for straight hairline. While for the allele of the father, we have 2 alleles for straight hairline. The next thing that we have to do is to cross the allele for widow's peak and the allele for straight hairline. So we have 1 capital letter W and 1 small letter w. Next, let us cross the allele for widow's peak and the allele for the straight hairline. One capital letter W and one small letter W. Next, we have allele for straight hairline and an allele for straight hairline. Therefore, we have two small letter W. And finally, an allele for a straight hairline and another allele for straight hairline. So we have another two small letter W. So let us interpret our answer. Our genotype are as follows. So we have two heterozygous trait and two homozygous recessive trait. So for our phenotype, if the genotype has an allele for a dominant trait and for a recessive trait, the dominant trait will be expressed while the recessive trait will be masked. Therefore, we have two widow's peak and we have two straight hairline.
Alright. Next problem. Polydactyly is a hereditary trait expressed as an extra finger on the hand or an extra toe on the foot. Given that both parents have heterozygous gene pair for the said trait, so this is polydactyly, what is the genotype of the mother? How about the father? How many kinds of gametes can the mother produce? How about the father? Perform a cross using the Punnett square. Give the genotypic ratio of the cross. Give the phenotypic ratio of the cross. You may pause the video to complete the answers. Alright, let us compare our answers. For number 1, the answer is one capital letter P and one small letter p. Why letter P? Take note, polydactyly is the dominant trait, therefore we will use letter P. Since polydactyly is a dominant trait and the genotype of the both parents is heterozygous, we will use one capital letter P and one small letter p to show that the parent has an allele for polydactyly and an allele for the normal numbers of the fingers and toes. Same as the genotype of the father. How many kinds of gametes can the mother produce? The answer is 2, an allele for polydactyly and an allele for the normal numbers of the fingers and toes. Same is true with the father. Then perform a cross using Punnett square and give the genotypic and the phenotypic ratio. Let us watch this video.
Polydactyly is a hereditary trait expressed as an extra finger on the hand or an extra toe on the foot. Given that both parents have heterozygous gene pair for the said trait, therefore, the genotype of the mother and the father should be written as capital letter P and small letter p. Why is that? Because they have heterozygous gene pair for the said trait. Remember that polydactyly is a condition wherein an individual has an extra finger on the hand or an extra toe on the foot. Since polydactyly is a dominant trait, we use letter P to represent the trait. For the normal numbers of the fingers and the toes, we will use small letter p. Next is we have to perform a cross using a Punnett square. So let's divide it into 3 columns and into 3 rows. So on the upper part, we will write the alleles of the father, and on the left side, we will write the alleles of the mother. The mother has a heterozygous gene pair for polydactyly. Therefore, we have an allele for polydactyly and an allele for normal numbers of the fingers and the toes. And same is true with the father. Then, the next thing that we have to do is to cross the allele for polydactyly and another allele for polydactyly. An allele for polydactyly and an allele for normal number of the fingers and toes. So we have one capital letter P and one small letter p. And we have an allele for normal numbers of the fingers and toes and an allele for polydactyly. Therefore, we have capital letter P and one small letter p. And finally, the last cross, we have an allele for normal numbers of toes and fingers, and the same here. So we have two small letter p. So let us interpret. So we have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. For our phenotype, we have polydactyly, so we have 3 polydactyly and 1 normal numbers of the fingers and the toes. Gregor Johann Mendel is an Augustinian friar or monk born in Bern, Austria. He is interested in investigating how individual traits were inherited. He wanted to find out whether both parents contributed equally to the traits of the offspring. He also wanted to know if the traits present in the offspring were produced by the blending of traits of the parents. Mendel chose the garden peas for his experiments. Before he started his experiments, Mendel first produced pure breeding plants. Mendel allowed his pea plants to self-pollinate for many generations until all offsprings had the same features as the parents, generation after generation. Inheritance can be explained in terms of unit determiners called genes. It is the gene, not the character, that is transmitted from one generation to the next. An allele is one of the two alternative forms of a gene. Genes in an individual appear in pairs. Gene pairs may be described as homozygous if they are made up of two identical alleles, or heterozygous if they are made up of two dissimilar alleles. When alleles for two contrasting traits are present in the same individual, the dominant trait is expressed, but the recessive trait is not. The two genes for a character separate or segregate during the formation of gametes. Hence, a gamete receives only one gene from every pair of genes. The genes for different characters are transmitted independently from one generation to the next. Thus, the inheritance of one character does not influence that of the other.
Okay, at this point, let us check if you have learned something from this video lesson. Number 1. In relation to your great-grandparents, to what generation of offspring do you belong? AP1, BF1, CF2, DF3. Number 2. Mendel experimented with garden peas. To ensure that all garden pea plants that he cultivates produce only yellow seed from generation to generation, he allows the garden pea to self-pollinate. When a homozygous yellow-seeded pea plant undergoes self-fertilization or self-pollination, what types of plants are produced? A. Pure breeding plants, B. Dominant plant for a particular trait, C. Recessive plant for a particular trait, D. Co-dominant traits among pea plants. Number 3. When two heterozygous animals mate, the resulting offspring is expected to exhibit a genotype ratio of A. 3:1, B. 1:2:1, letter C. 1:2:1:2, Letter D, 9:3:3:1. Number 4. Today, dominant and recessive genes are represented by a capital and a small letter, respectively. Which of the following symbols represent a pair of homozygous recessive genes? A, B, C, and D. And number 5. If a test cross results in all heterozygous offspring, then A. One parent must be heterozygous for the dominant trait. B. One parent must be homozygous for the dominant trait. C. Neither parent has received the trait. D. Both parent must be heterozygous. Let us have the answers. Number 1, letter D. Number 2, letter A. Number 3, letter B. Number 4, letter C. And number 5, it's letter B.